Hello students, this is Dr. Faizan Mirza. We are discussing control of ventilation, the central and peripheral chemoreceptors, how they function in your respiratory system. So here, what I'm discussing here with you are the central chemoreceptors. I'm using this green color to highlight the central chemoreceptors. Central means they are present in the central nervous system. This is your brainstem. In the brainstem, there is a region called pons and the medulla oblongata, and this region is your central chemoreceptors. So this region is having the central chemoreceptors, the respiratory center. So the respiratory center I am drawing in green here. If we look microscopically, so this green region is made up of these kind of cells that form synapses with each other, and at the other end they are having some neurons, and those neurons go and make the synapse with these, and these neurons, the purple ones, are the ones which are the central chemoreceptors. So the central chemoreceptors have dendrites which detect this yellow substance ahead of this neuron. So around these dendrites, these are in the tissue fluid, this is this yellow substance. The yellow substance is the carbon dioxide. So the carbon dioxide value is detected here. So how is it detecting it? For example, this is respiring, it releases carbon dioxide goes into the tissue fluid. From the tissue fluid, it goes into the blood. The blood takes the carbon dioxide away. So that's the something that normally happens, that all the carbon dioxide which is released by these cells because they are actively respiring goes to the tissue fluid and goes to the blood, and the blood takes that carbon dioxide away. What's the cause of the carbon dioxide going? The diffusion gradient exists. There's less carbon dioxide in blood and more carbon dioxide in tissue fluid, so the carbon dioxide goes out into the blood and away. At this time, action potentials will be generated in this neuron and less neurotransmitter will be released, so these respiratory centers will be stimulated less. As a result, the respiratory rate will be controlled. The person will keep on inhaling and exhaling at a normal rate because there is not a lot of carbon dioxide being built up inside the body. But if the PCO2 increases, now when PCO2 increases, it means that your blood is already having a high carbon dioxide concentration. Now when this blood comes with high carbon dioxide concentration, since it's already having a lot of carbon dioxide, the carbon dioxide being produced by these cells comes into the tissue fluid here, but it cannot diffuse very well into the blood because the blood itself is having a high carbon dioxide. So the diffusion gradient is not steep now. As a result, the carbon dioxide accumulates around these cells which are your central chemoreceptors. This is the interstitial fluid or tissue fluid. At this time, these central chemoreceptors end up firing a lot of action potentials. The frequency increases, the impulses are sent more frequently, and more of the neurotransmitter is released at the synapse. This in turn causes the stimulation of these cells which are your respiratory center in the pons and the medulla oblongata. When these respiratory centers are activated, they increase the breathing rate. So when the breathing rate increases, this allows more carbon dioxide to be sent out of the body from the lungs, so you exhale more frequently. So the respiratory rate increases. Now we know that carbon dioxide can combine with water to form H2CO3, and H2CO3 can then dissociate to form H plus and HCO3 minus ion. We know that high PCO2 actually means low pH, because a low pH is because of a high H plus concentration. As a result, your central chemoreceptors can detect both. They can they can detect and monitor low pH and a high PCO2 both, but they cannot detect the changes in the oxygen levels in the interstitial fluid or in the blood. So what the central chemoreceptors are doing, they change their breathing rate depending on the carbon dioxide present around them. Now, what about the peripheral chemoreceptors? How the peripheral chemoreceptors are working? Peripheral chemoreceptors work by a different mechanism. I have taken this diagram from a different source. So you can see this is a human heart. The human heart arches up the aorta. Ahead of aorta, the blood vessels go. You have the carotid artery, right and left carotid artery. They form a carotid sinus, and the carotid sinus allow exterior and the interior branches to go into the brain towards the head and neck region. Now, here inside the sinus there are these baroreceptors, and the baroreceptors are present here as well. So these baroreceptors monitor the changes in the blood pressure. And right outside these baroreceptors in the carotid arch, there is you can say that here there is this region. These are the chemoreceptors present outside this tissue, right here and right here. These are called as the carotid body and aortic body respectively. So the baroreceptors and the carotid body and the baroreceptors and the aortic body, they both are in close proximity. So this is the anatomical situation of how the baroreceptors are in contact with the chemoreceptors in the aortic body, and the baroreceptors are in close contact with the carotid body as well. PO2, which is how much oxygen is there, how much carbon dioxide is there, and what's the pH, they all are monitored by these chemoreceptors. The chemoreceptors are in the carotid body and the aortic body, as already described. So if we magnify this region, how exactly the carotid body is working with the you can say the baroreceptors. So the carotid body here is receiving a continuous blood supply, and this carotid body is made up of tiny cells. So all these cells are receiving capillary blood from here, so they actually can sense how much oxygen and carbon dioxide is there. The same goes with the aortic body as well. So here this branches of these capillaries, they provide these cells which are of the aortic body here with fresh blood. So they are constantly noting down the PO2, PCO2 or pH there. This is the perfusion in this carotid body, 2 liter per minute per 100 gram. That's enormous blood supply for 2 minutes. So that actually tells you how well the perfusion is there. So they keep on monitoring the amount of carbon dioxide, oxygen, and pH in blood. Now how these work? They also work by the diffusion gradient. How they work by diffusion gradient? This cell, this yellow cell, is the same cell that's present here. So the carotid body and the aortic body is actually having glomus cells. What are glomus cells? Glomus cells are specialized cells which can release neurotransmitters when stimulated. So this yellow circle is actually the glomus cell coming from the aortic body or the carotid body. The blood supply next to it, for example, if there was a lot of oxygen here, so the oxygen which is coming from the capillary blood, it diffuses out into the tissue fluid and it comes to the glomus cell. The glomus cell gets all the oxygen and it uses up that oxygen. Now let's suppose if there is less oxygen coming here in the blood, it causes less oxygen to move into the glomus cell. The glomus cell depolarizes. When it depolarizes, it ends up releasing the neurotransmitter vesicles from inside the cells. And when the neurotransmitter vesicles are released across the synapse, the axon terminal of the next neuron becomes stimulated and it ends up transmitting the impulse ahead. So the nerve impulse goes ahead and the breathing centers are stimulated and the rate and depth of breathing changes. So this is how oxygen is monitored by the glomus cell. Now what happens with carbon dioxide? The very same condition happens with carbon dioxide. Since this cell is respiring, it will release a carbon dioxide, a lot of carbon dioxide. The blood must have low carbon dioxide concentration. So all carbon dioxide that forms here should go diffuse, should go out into the blood, should diffuse here, and the blood should take that away. But for example, if the blood was already having a high carbon dioxide concentration, all the carbon dioxide that the glomus cell produced could not pass out into the blood because the diffusion gradient was not there. The blood was already having a high carbon dioxide, more orange circles here, more carbon dioxide here, and less here. So the carbon dioxide could not go. Since the carbon dioxide fails to diffuse out, again this excess of carbon dioxide build up in the glomus cell. Since it could not diffuse out, it again caused the depolarization to occur. So the vesicles, they release neurotransmitter here. The axon terminal is stimulated, the breathing centers are stimulated, and the rate and depth of the breathing is altered. We can recall how carbon dioxide combined with water to form H2CO3 and how H2CO3 is associated to form H plus and HCO3 minus ion. So H plus can also be detected in the same way, which means the pH can also be detected in the same way. All the H plus forming here goes to the blood must have a low H plus concentration for the diffusion gradient to exist. So if the diffusion gradient exists and there is less H plus concentration here, more H plus formings here, the H plus will go into the blood and the blood will take the H plus away. But if the blood is already having a lot of H plus, which means there is a low pH, a lot of H plus in blood. A lot of H plus in blood means that the glomus cell will not unable to clear off its own H plus concentration. The H plus will stay here accumulated. When the H plus is staying here, the cell depolarizes, it pushes its neurotransmitter vesicles, and exocytosis allow the vesicles to release the neurotransmitter into the synapse. The axon terminal gets stimulated, the nerve impulses are sent to the breathing center, and then the rate and depth of breathing is altered. That's it from my side. Thank you so much.